Really my pleasure today to introduce both of our speakers. Before we get started, if you haven't read the slide already, I just remind everybody that if you do have questions during or after the seminars, feel free to raise your hand and I can unmute you and allow you to ask the question yourself, or type your question into the Q&A and I can ask it for you at the end of the seminar. Beyond that, I'm really excited to now introduce Kasit Chatsirisupachai. Sorry. Kasit did his bachelor's and master's at Mahidol University in Bangkok, and then moved to Liverpool in the UK to do his PhD with João Pedro de Majales, where he worked on the relationship between aging and cancer and published some really cool papers. And in 2022, he received an EMBO long-term postdoctoral fellowship and moved to Heidelberg at EMBL, where he is now working with Arno Krebs. And just recently had a really, really cool work published that he's going to tell us about today. So without further ado, Kasit, please take it away.
Right. So thank you very much. It's really my great pleasure to be here. And actually, I have to say that I've watched several cool talks from this seminar series, and I really enjoyed them. So very, very nice to be able to talk to you about our latest study. As Jackson just mentioned, we just published it actually last month. So the story I'm going to tell you is about the occupancy of RNA polymerase II as the promoter of mouse and Drosophila, two of the most widely used model organisms. Right. So I think actually this audience don't need any introduction to this, but just in case, this is just a very simplified model of transcription. Usually we have these regulatory elements, such as enhancer and promoter on the DNA. And promoter is like just sitting in front of the gene, and that is where the transcription happens. Enhancer can be located megabase away, and usually it works by the binding of transcription factors to the enhancer elements, which also recruit other types of protein like cofactor, for example. And together, somehow this signal got translated into the promoter, and that helped recruiting general transcription factors to form the pre-initiation complex, a huge protein complex that also include the machinery for transcription that is called RNA polymerase II, which can start transcribing the gene. And if we zoom into this promoter region, there are several steps at the initiation of the transcription as well. So typically in let's say many promoters, RNA polymerase II would start transcribing for about 50 nucleotides, the process that we call transcription initiation. Then it's gonna stop. So this is what we call pausing by the involvement of some protein factors here. So pausing itself is important as a quality control step. It has also been proposed as a regulatory step, waiting for other signal to come and start transcribing further. For example, in the case of stress response gene, right? And after that, Pol II would be released into the gene body and to go into the productive elongation. However, not all RNA Pol II would go in this direction into elongation. The other important phase of the post-Pol II here is to leave the promoter by the process that we know as early termination, such as by the integrated protein involvement here. So in this slide, we'd like to highlight you the phase of RNA Pol II initiation, pausing, and leaving the promoter either by elongation or early termination. So we know that the transcription machineries are actually highly conserved across metazone. For example, in this nice preprint here, you look at the conservation of negative elongation factors, the protein complex involved in RNA Pol II pausing. And as you can see here, it is highly conserved from, for example, Drosophila up to mammalian system like mouse or human. And together with that, you can also see the Pol II pausing signal here. I think it is from ProSeq data. And you can see the shape of protein signal is kind of very similar between the two species. And the pausing site is around 50, 60 nucleotide. So by this, we know that transcription machineries are highly conserved. So probably the transcription itself from the both model species should be fairly similar. But that is unfortunately not the case because there are the evidence from live cell imaging of transcription. For example, if you track the transcription in real time in single cell and single promoter, on the x-axis here is time, and on the y-axis is the, let's say, RNA production, right? So you would expect in the case of Drosophila to see that the promoter would spend roughly the same time at an inactive state as an active state. This is in an order of minutes. On the other hand, if we look at mammalian system like mouse or human cell lines, this is going to be totally different because most of the time the promoter would spend in an inactive period and interspersed by the short bursts of active transcription. And you think about this and look across cell population, not just only a single cell, and then you take a snapshot at a certain time period. So what you expect to see is that in the majority of the cells in the population, in the case of Drosophila, would have RNA Pol II binding at the promoter during an active state of transcription. On the other hand, if you do the same thing in mouse, what you would expect to see is that RNA Pol II would bind at a very low frequency in the cell population. This is, again, if we're looking at a single promoter across cell population. That is the expectation, but in reality, what we see, again, this protein signal, you will see fairly similar signal between the two species. Protein is measure the engaged polymerase, right, by capturing this nascent transcript. But actually, this is the average from the population of over 1 million of cells. So to help you think about this more clearly, if you think about technique like ChIP-seq or PRO-seq, what we are measuring itself is the bound fraction of DNA. For example, we use antibody in the case of ChIP-seq, or the nascent transcript in the case of PRO-seq, and then we enrich only those bound molecules before sequencing. So what it's going to give you is the enrichment over the genomic location, right? For example, in this case, you know that in this location, there are more Pol II than the other location. But we don't know, we know that you can also give the fold chain. For example, this has twofold more than this location, but we don't really know the percentage of the cells in the population that is bound by RNA Pol II. There's something we actually blind to. On the other hand, if we look into a single molecule space, this is the different principle, right? Because we sequence everything regardless of the binding state, don't enrich anything, and then we sort out afterward. So in this case, we can put on the number. For example, this is the example again, and you can see on the left-hand side, 100% bound molecule, meaning that every cells in the population here have Pol II bind at this certain location. On the other hand, on the right-hand side, just only 50% bound. So just want to highlight we are trying to move from bound assay to a single molecule assay. And perhaps we can ask the question that, for example, is there any difference in the frequency of Pol II occupancy between the two species? And also, if that is the case, what would be the mechanisms that controlling these differences? Right. So single molecule assay that we use for single molecule footprinting, I think many of you know the principle already, but for some of you that probably this is the first time you've heard about it, the idea is very simple. We use DNA methyltransferase, specifically here is the CpG and GPG enzyme, to methylate accessible cytosine, meaning that if the cytosines are protected either by nucleosome here or proteins of interest like pre-initiation complex or an RNA Pol II, they're not going to be methylated. I just want to walk you through the average plot of the single molecule footprinting. This was done by Arno, my supervisor, in his postdoc paper. So he looked at Drosophila promoter, and in this case is the promoter of Hitchcock protein 70. And what you are looking here on the x-axis is the location in the genome. And on the y-axis is the average methylation level. No, it's actually the inverse of the methylation, sorry. So meaning that on the top here, 100% meaning that there's no methylation or is highly protected. And on the bottom here, if you have a 0% of SMF signal, it corresponds to 100% methylation or highly accessible cytosine. And each dot you're looking here correspond to the cytosine location and the inverse methylation level of the cytosine in that location. As you can see here, this is the nice footprint of pre-initiation complex and RNA polymerase II, as well as the large footprint of nucleosome. So the bar average is cool, but the beauty of the technique itself is that we can go into single molecule resolution and look at the methylation within each DNA molecule. So what you are looking here is a stack of about 1,200 DNA molecule. On the x-axis, again, this is the position in the genome, and each column here corresponds to the cytosine location. And what you're looking here is the methylation status of the cytosine. So if they're black, they are protected, or something is binding there. And if they are accessible, is shown in gray. So when you sort this molecule out basically by clustering, what you are looking here is that in this particular promoter, 35% of the time, RNA Pol II would bind at this pausing site. So after that, the question is that how can we do the same thing, look at the Pol II at the mouse promoter instead of Drosophila? And the short answer is yes. As you can see here, this is the example. In this case, you can also see this pre-initiation complex footprint and RNA Pol II footprint here, which correspond to about 12% in the cell population that have Pol II bind at this location. So a single example, can we do it more globally? Now we compare this average of the highly active promoters. So this is top 5% highly active promoter, which correspond to roughly 1,000 promoters for each of the species. And in Drosophila, you can see a nice footprint of pause polymerase here at the pausing site, but this is completely absent at the mouse promoters. So this is very surprising at the beginning. We wonder why. And again, because we can go into single molecule, we can give some number. So in the case of Drosophila, Pol II would bind roughly about 15% in the cell population. And this is going to be reduced to only 3% at mouse promoters, meaning that they are five-fold different in terms of Pol II occupancy here between the two species. So we reason that Pol II that we are capturing here at the pausing site, we know that there is 5 times lower at mouse promoter comparing to Drosophila promoters. This could be coming from the ratio between the incoming Pol II, meaning that in mouse promoter, probably there are lower initiation rate at the promoters, or probably it can coming from a higher turnover rate or Pol II leaving the promoter, right? And here we cannot distinguish between the two phases that I introduced earlier, elongation or early termination. So this different number here could coming from the different incoming Pol II or different in the outgoing Pol II, if you like. And we collaborate with Nacho. So he transformed this intuition into an equation that is very simple. And as you can see, we have these only 3 parameters. Q correspond to Pol II occupancy that we can capture using single molecule footprinting. And then if we can quantify either the initiation rate or the turnover rate, we could calculate back the other remaining rate. So that's exactly what we did. We inhibit new transcription initiation using small molecule triplet, many of you know probably. And then we track the Pol II at the pausing sites over time using PRO-seq here to capture nascent transcript. So if we don't have new incoming Pol II, and then we track the signal of Pol II over time, we can look at the rate of Pol II leaving the promoter. And to cut a long story short, what we found is that turnover rate is higher at mouse promoter when comparing to Drosophila promoters. As you can see that in mouse promoter in red box here, the signal drop is sharper than in Drosophila, the blue box. So we can calculate, of course, that is about 2.2 times higher in turnover rate at the mouse promoter than in Drosophila promoters. And as you see, 2.2 do not match with 5. So we basically substitute this into the equation and calculate again. It's about 2.24 lower in the transcription initiation rate at mouse promoter. And on the right-hand side, we plot this inverse of transcription initiation rate, or you can interpret it as the waiting time until the new transcription starts, which in the case of Drosophila is roughly 20-30 minutes here, and in the mouse can go up to an hour or even more than that. So again, I would like to highlight this is correspond to what people see when they do live imaging transcription, that in mouse promoter, there are longer waiting time until the new transcription happening. Right, to summary, we showed by using single molecule footprinting that Pol II occupancy is lower at the mouse promoter comparing to Drosophila promoter. And that is coming from the two main reasons: lower transcription initiation rate at mouse promoters and higher Pol II turnover rates. And this paper was just published. If you are interested in it, I left out a lot of detail, but if you're interested in it, just published like last month, and we have a nice Q cover here. So finally, I'd like to thank people in Krebs Lab, especially Arno, who took me on board to the project. I would like to thank past members, especially Christine. She actually initiated the project and before I joined the lab. And our fantastic collaborator, Nacho. And finally, I'd like to thank the funding source that allowed me to stay in Heidelberg, EMBO, and later on DFG. And happy to take questions. Thanks.
Great, Kasit, that was really great. Do you think that, I guess, do you have any hypothesis as to why this difference has arisen in mammalian cells versus insect cells?
No, that's very, very cool question. And I've got asked this question several times when I present the story. So we don't really have a definite answer. I think there might be something related to a biochemical rate inherently into different species. As you know, this could also correlate with developmental timing, lifespan. That's what people have been showing. So in short, I don't think I have an answer to this, but it would be very interesting to look into this.
I guess along those lines, Azim Khan asks in the Q&A, have you observed what causes the higher transcription rate for Drosophila?
No, I mean, it would be cool to. So in this project, we didn't really measure transcription rate, right? I mean, it would be great to measure it, but we just cannot think of the way to do it genome-wide in
Right.
both species that is doable. Yeah, but definitely it would complete the cycle if we, from the prediction, right? We would go and try to measure the transcription initiation rate. So, no.
I was curious about with triptolide. Do you see the same dynamics in Drosophila cells as with mouse cells? Like, does Pol II get degraded as rapidly in Drosophila as it does in mouse? Or
Graded, I don't think we measured the degradation, but the time point that we look into triptolide is very short. We start to see the effect like, very early on, like 2.5 minutes. Sorry, I should have been more explicit on it. And we measure only until 20 minutes. And as you can see, most of Pol II this, again, it highly active promoter in mouse is already gone. But in Drosophila, there's still some fraction there.
Okay, so then let's see, a couple more questions in the Q&A, and then we'll move on. Felix asks, can you differentiate the pre-initiation complex and fragile nucleosomes in the promoter regions? And do you think there's a difference in fragile nucleosome content between mouse and Drosophila? Thank you for the very thematic question, Felix.
Yeah, that's all great questions. I mean, we never look at fragile nucleosome. So the idea is that I don't know if we can try to disentangle fragile nucleosome by using footprinting data alone. I mean, I don't have a good answer to this. We never try to look into it more detail. Maybe yes. But yeah, thanks a lot for great questions.
And then I'll ask one more quick one, potentially. Maria asks, are there any sequence differences in the pause sites in mouse and Drosophila? And is it just that pausing is better in flies?
That's a good question. At the pausing site cell, probably not really. But one thing that we do observe, I think people know already, is that in the mouse promoters, it's just promoter region, not necessary to be only the pausing site, but the promoter region in mouse is highly GC. No, it's like the high CPG islands. That's not in Drosophila. So yeah, that could be something that influenced, but we didn't follow this up.
Right.
Yeah.
All right. Well, you've got several more questions in the Q&A, but in the interest of time, I'll let you go ahead and answer those as we move on to David Suter, our next speaker.